Every minute, millions of red blood cells circulate through your vascular system in continuous, high-speed transit. To understand what actually moves through your veins, we must look past the cell itself, penetrating the membrane to observe its molecular core. This is a 3D model of a hemoglobin molecule. By stripping away the protein structure, we expose the engine of the cell, 4 central iron atoms locked within the heme group. In 1936, chemist Linus Pauling and his colleague Charles Coriell measured the magnetic properties of this specific iron atom. Their data revealed a strange discrepancy in how human blood interacts with magnetic fields. Notice these electron paths. Deoxygenated venous blood has 4 unpaired electrons, making it paramagnetic, attracted to magnetic fields. But when oxygen binds, electrons pair up. Arterial blood becomes diamagnetic, repelling magnetic fields. This creates a massive 20% swing in magnetic susceptibility. The iron in human blood switches between 2 distinct electromagnetic states with every single heartbeat. The circulatory system functions as a continuous, oscillating electromagnetic circuit. Human taxonomy has traditionally categorized populations based on superficial morphological traits, primarily the pigmentation of the skin. Conventional science often describes melanin as a cutaneous filter, a natural sunscreen designed to block ultraviolet radiation at the surface. If melanin is only a UV filter, it leaves a major biological question unanswered. Why does the genetic receptor that controls it, MC1R, also dictate the functionality of deep internal structures, like the inner ear and the density of our bones? MC1R functionality determines the organism's degree of environmental fitness across electromagnetic, oxidative, structural, and neurological domains. Melanin serves as a universal survival molecule designed to regulate the body's internal electromagnetic coherence. Visible skin color is the byproduct of this systemic regulation. Red blood cells are manufactured inside the dark core of human bones, the marrow, rather than in a dedicated organ exposed to the surface environment. Researchers at the Zahn Laboratory at Harvard University tracked how blood stem cells migrated into bone marrow as terrestrial vertebrates moved onto land. This process is hematopoiesis. Hematopoiesis is the biological mechanism where stem cells generate the entire supply of circulating red blood cells. The Harvard study showed that these stem cells required extreme radiation shielding to survive on land. They were only able to persist because neural crest melanocytes formed a protective, light-absorbing barrier directly over them. Without this specific melanocyte barrier, ambient ultraviolet radiation and oxidative stress rapidly penetrate the tissue and destroy the blood stem cells. Melanocyte protection is the structural prerequisite for hematopoiesis. The blood factory exists because the melanocyte built the shelter. Because of their iron state, red blood cells physically move and migrate in response to magnetic field gradients. As these cells cycle through the body, their continuous paramagnetic to diamagnetic switching constitutes a literal oscillation of electrical charge within the blood plasma. This charge oscillation is the exact biophysical mechanism utilized by modern medical imaging. Functional magnetic resonance imaging, or fMRI, maps brain activity entirely by tracking what is known as the bold signal. These yellow activation zones represent the bold signal, blood oxygen level dependent signal. It measures the ratio of oxygenated to deoxygenated blood. The fMRI scanner is explicitly calibrated to detect the magnetic state change of iron as it delivers oxygen to neural tissue. Every time an fMRI maps human brain activity, it is tracking the electromagnetic oscillation of iron. The entire system operates as a unified frequency network. Melanin is the chemical regulator that safely chelates, or binds, this oscillating iron to keep the system stable. The ancestral state of the MC1R gene, the wild type, produces eumelanin. Eumelanin securely locks the iron atom away, preventing it from damaging the surrounding cellular fluid. When an individual carries loss-of-function MC1R variants, the body defaults to producing structurally unstable pheomelanin, which drops the free iron atom directly into the cell under stress. This initiates the Fenton reaction. The free iron reacts with hydrogen peroxide to generate a cascade of toxic hydroxyl radicals, destroying nearby cellular structures. In the brain, neuromelanin normally holds iron securely, but pheomelanin-based neuroneuramin releases it, driving a 2 to 3 times higher risk of Parkinson's disease in variant carriers. In the inner ear, the absence of melanocytes in the stria vascularis destroys the endocochlear potential required for hearing. This auditory collapse is thoroughly documented across species. The congenital deafness observed in blue-eyed animals, like the Dalmatian, links a lack of external pigment to the internal absence of cochlear melanocytes. MC1R loss of function triggers a systemic degradation of the body's electromagnetic architecture, leading to the predictable structural and neurological failures observed in variant carriers. Evidence of this altered architecture is visible on your own wrist. Some populations exhibit visibly blue veins, while others exhibit green veins. The standard clinical explanation claims this is an optical illusion caused by skin melanin acting as a light filter. But this fails to account for individuals with light skin who still present with green veins. Blood inside a closed venous system exists in a specific, unoxidized chemical state. Because the vein wall is translucent, the color you see is the actual spectral signature of the blood, projecting its wavelength through the tissue. This optical difference correlates with the RHD gene deletion, a missing protein on the red blood cell membrane. This deletion is concentrated in the same populations carrying the MC1R loss-of-function mutation. Blood plasma is an ionized medium. By altering both the melanin protection governed by MC1R and the red blood cell membrane architecture, the biophysical composition and the spectral output of the plasma itself physically shift. Vein color provides direct, naked-eye evidence of the spectral frequency inside the vessel, reflecting the current state of charge oscillation and biophysical health. The biological record demonstrates that melanin's regulatory function is a consistent principle across every kingdom of life. Inside the extreme radiation environment of the Chernobyl nuclear reactor, specific species of melanized fungi actively thrive. These fungi utilize their melanin to capture gamma radiation and convert it directly into chemical energy, proving melanin functions as an ancestral energy transducer. This survival mechanism contrasts sharply with what happens during evolutionary depigmentation in lightless environments. The blind cave fish, Astyadax mexicanus, lost its functional MC1R gene after 30,000 years in lightless aquatic caves. This resulted in the total loss of both its melanin and its visual structures. Whether an organism is a fungus inside a nuclear reactor or a fish in a pitch-black cave, its structural integrity is dictated by the presence or absence of melanin functionality. Between 10 and 40,000 years ago, during the late Pleistocene, human populations found themselves trapped in catastrophic low ultraviolet ice age environments. Human depigmentation events were severe biological trade-offs necessary to survive these freezing, low-light conditions, mirroring the adaptations of the cavefish. SLC24A5 is a gene that regulates calcium and sodium exchange to control melanin biosynthesis. These mutations did not create new subspecies or different races of humans. As this diagram shows, they resulted in populations operating with a compromised electromagnetic architecture, stripped of the ancestral iron chelating protection that sustained their cellular coherence. It is time to abandon the obsolete morphological container of species. We must classify human biology by the functional mechanism inside, the state of our electromagnetic coherence.